When we think about neurons, it's like Broadway at 5 p.m. here in New York. So imagine if all of a sudden we put big stones in the middle. So of course they can go around, but that's like possibility of having crashes are going to be high, and it's going to slow things down. So at the beginning you have a slow function, and neurotransmission or communication among neurons is key for the functioning of your brain.
So that cleaning process have a name?
Yeah, so the cleaning is called autophagy. So auto means yourself, and phagy means eating. It's about eating the garbage, and the beauty is that not only you eliminate that garbage, so this autophagy happens in lysosomes. They are the most perfect recycling mechanism in the world, because every single day they take all the garbage of your cell, they chop it in little parts like amino acids or sugars or lipids, and then that's being recycled so the cell can use it.
You're listening to the Neurochemistry Podcast, presented to you by the International Society for Neurochemistry. Thanks for tuning in. Let's get started.
Hello, everyone. Thanks for being here on the Neurochemistry Podcast, where neurochemistry is for everyone. I'm Will Sanchez, and today our special guest is Maria Cuervo. She is a distinguished professor at the Albert Einstein College of Medicine. Also, thank you very much,
Thank you for
being
having
here.
me, William.
Oh, it's a great pleasure. So let's talk a little bit about what you are doing in your lab. So what are your main interests in your lab?
So our main interest is on cleaning. I think I inherited that from my mom. She was obsessed about cleaning, and that kind of came into my research. And what we are interested in cleaning is actually the cells in your brain. As you probably know, every day you produce toxic products because of all the stress, and everybody knows about free radicals and they cause damage. So you need to make sure that anything that is damaged or is not in good conditions inside the million cells that you have in the body has to be cleaned every day. So we are very interested in understanding how that cleaning happens, and then also why it doesn't happen as we get old, and also in diseases of aging. That is very appropriate for this interview because of the very severe high incidence of neurodegeneration in our society these days. Diseases like Alzheimer and Parkinson, we are convinced that they start in part because you don't clean well your neurons.
Exactly. So for people that are just hearing us and are attending this podcast, so that cleaning process have a name, a term?
Yeah, so the cleaning is called autophagy. So auto means yourself, and phagy means eating, but it's not about cannibalism or anything like that. It's about eating the garbage, and the beauty is that not only you eliminate that garbage, but you recycle it. So this autophagy happens in kind of containers that are called lysosomes. And people always study them. When you study them in the school, it's like, oh, the garbage containers of the cells. But my mission in life is to make them more glamorous, because they are not only garbage containers, they are the most perfect recycling mechanism in the world. Because every single day, they take all the garbage of your cell, they chop it in little parts like amino acids or sugars or lipids, and then that's being recycled so the cell can use it to generate new proteins or to obtain energy to keep going. So that's very coordinated and very important. So no more garbage containers, mostly recycling.
So the balance between the degradation, and also to use those part of the molecules that the system degrade can be used. So the balance is important in our brain.
The balance is really important in our brain because in most cases, and that's why we think this system is important, you utilize it for cleaning, but like for all of us, right, you need a motivation to clean. It's like you clean because you are going to host somebody in your house, and then it's like, oh, it has to look good. So the motivation that the cells have to start cleaning is that they get energy out of this recycling. So if you are not eating continuously, basically your cell every day, all your cells will activate autophagy, because it's like, well, this person is not giving me the energy that I need, so let me see what I can eliminate, and then I can recycle. And it's like, you go for the things that are damaged, because imagine that you go to your winter house and then the heat is not working, but you have a beautiful fireplace and you can say, oh, I can really make a fire, and you cannot go outside. So you're not going to go for your most beautiful chair. You're going to go for this one that is not so good, or this one all. And that's what the cell does. It looks like what is wrong, what is not good, so I can utilize it for energy for the cell. And that balance between nutrition and what we eat and autophagy is what it makes it so relevant, because there are many ways that we can activate this cleaning if we will not be eating 24 hours a day.
Right. So let's imagine that one of our chairs is that protein. This one is that I'm sitting right now. It's an old one, so it's not working that well, and I need that wood for my fireplace. So the brain is clever enough, the system, to to choose what is working not that good, and to first is use occupying space in my living room, but also I can use it for something more important, right? What happened in our brain when our cleaning system is not working that well?
Yeah, so there are several possibilities, but one of them is that many times there was this question that is like, how does the brain differentiate between the broken chair and the functional one, right? Because it's like you just have the garbage container there, and how are you bringing it? So part of what my lab has done through many years is to try to figure out how does the cell discriminate. And what we know now is that there are these chaperones that is really similar to the old concept of chaperone. You guys are too young, but when I was young, if you go to the movies, you have a chaperone, so you don't do anything with your boyfriend that you shouldn't be doing. So the chaperone does the same. It makes sure that the proteins behave, and behaving for a protein is to be in the right place at the right time and in the right conformation, right? Like no damage, all beautiful there. So if it's in the wrong place, or if it's just like broken, the chaperone identify and say, okay, this has to go out. So that gives the selectivity that only the chair that is broken, the chaperone is gonna go for it. And then it bring it to this recycling container. So there is this process of selectivity, and that's one of the things that we've been investigating for many years. But going back to this idea, what happened, you can have two problems. Once you lose the selectivity, so then you are sending everything to your garbage container, and that's not good either. You don't want to sacrifice your perfect chair. And so there has to be this very tight regulation of what you are eliminating. But what happens more as we get old is that the chaperone is still doing the police work. Is like, okay, this is good, this is bad. Unfortunately, the recycling containers are not ready to receive. And then to picture what will happen in your brain, or in the cells in your brain, just think when people like in the cleaning system, for example, in airports, if they go on a strike, if they are not cleaning for one day, I mean, you all have seen the pictures of garbage accumulating all over. So that's what happened. And it happens first inside your neurons, the cells of your brain. They start accumulating proteins, they start accumulating damage. And then what we have learned through the time is that there is a point that when you have too much garbage inside these neurons, they have to save themselves. There are very important cells, and they are like, I cannot clean inside, I'm just gonna throw it through the window. And that's what they do. Basically, the neurons start throwing this garbage through different cellular mechanisms, but they send it outside. And that contributes to propagation of disease, because now not only you have this poor neuron that was not in good condition, but all this garbage is going to be floating in the streets and the highways in your brain.
Nobody take responsibility to clean that.
So initially you have some systems outside your neurons, and that's why the neurons probably they say, well, there is people cleaning outside, so let's throw it. So let's do that. And that works for a while, but then with the time, when you have everybody throwing the garbage outside, those systems, the lymph become overwhelmed, and basically you cannot clean it anymore. And I know it sounds silly to be talking here about broken chairs and sending garbage, but the basis for many neurodegenerative conditions, like we mentioned Alzheimer, Parkinson, is a protein that misbehaves. Either you have a mutation, or it has like a modification that make it unwanted. And if you think about those patients, they don't have Alzheimer when they are 10 years old or 20 years old. They really have Alzheimer as they get old, when they already have that mutant or that misbehaving protein. So the reason why the brain is functioning within in part is because the brain is still able to clean this mutant protein. So as far as you are able to eliminate it, it will be okay. It's like at least you are doing this periodically. It's not a protein that you want to have around. Fortunately, you have that mutation, but you can eliminate it. But as you get old, and that's why aging is the major risk factor for all these diseases, we have seen that even with physiological aging, as you get old, cleaning is going to go down. So you are producing the same amount of the mutant protein, the same amount of broken chairs, but now you don't have anybody to eliminate them. And that's what built up, and everybody have heard about amyloid and tau tangles. So these are this accumulation of this misbehaving protein that then it can become like a chew gum, right? Because if a protein remains in the cell in a not very good conformation, it's very sticky. So anything passing by is gonna get stuck there. So then you
Gonna
build
grow and grow
you
like a
exactly
pile of garbage
like
gonna grow
pile
and
like
grow.
like
So
the
that
snow
become
rolling, right?
toxic for the brain and for the society to have that, like in that parallel. I love that analogy. It's not silly. It's a really good one. So it gotta be toxic for the environment. In this case, the
Correct.
cellular environment in our brain.
But eventually it goes even outside, right? Because it's like you start in the cell. Now all the cells are throwing this amyloid outside. And that's really going to make things very difficult.
Well, let me ask you something. So it when these aggregates of proteins they become, let's say, toxic uh, inside and outside the cells. So that affects in what, in which way it affects, like communications. It affects in the life cycling of the surrounding cells.
So it depends of the severity of the aggregates, and this is always progressive. So initially, it will be mostly the function of the neuron itself. So the neuron will not die, but you just imagine. I mean, when we think about neurons, you have neurons that they are as long as your body, right? They have to go from your brain to your toe, or from your spine to your toe, and in the brain, they have to be very interconnected. So we all have seen movies of things how they move inside neurons, and things is like a highway, or it's like Broadway at 5 p.m. here in New York. So basically it's like cars are going in all the directions. So imagine if all of a sudden we put big stones in the middle. So of course they can go around, but that's like possibility of having crashes are going to be high, and it's going to slow things down. So at the beginning you have a slow function, and neurotransmission or communication among neurons is key for the functioning of your brain. So that start deteriorating. The connections between the neurons that are called the synapses, they start getting also full of garbage. They don't function well, and then they start retracting. It's like, I'm not going to connect to the next one because I'm just going to make a mess.
When you're going to no, I'm not gonna take that way because it's too crowded. No.
So let's go to the other.
Got it.
So that's that. And then eventually when the cell, I mean, we have to think on the cells in the brain, the neurons are interconnected, not only with other neurons, but with supporting cells that are called glia. So once they become isolated because they are full of all these things, they eventually die because they don't have the factors that they need. They don't have like same thing that when we don't have social interactions, and also because they are full of all this garbage. And the problem is that when they die, they keep contaminating the environment, right? Because now you have this dead cell that eventually will be eliminated, but is so full of these aggregates that it's going to take longer than a cell that is not full of garbage, obviously. So that contributes to gradual neuronal loss. And sometimes people say, well, we have so many neurons that we don't use in the brain, right? But in a way, that's a misconception, because having neurons doesn't mean that they are properly connected. So you need to maintain that connectivity. And you create that connectivity through your life, and all of a sudden you lose that link in between these two parts. So that's going to affect your brain. Even you have some reserve ones that hopefully you might be able to bring to the brain.
I love that parallel because okay, it's good. Like it's important, the connections between each other, but as you pointed out, the functional aspects of some neurons can be different from others. So if I have, like, I don't know, a fire department, I have a hospital here, it's not the same if I replace this with and eventually communicate a fire department, and it's not the same to having the hospital. So you need to have the functions, communications, and the role of each cell. So it's good you touch up about, and probably this is going to be important to context people about other cells than neurons, right? You touched something about the maintaining cells, if you will, in the brain. How can you like elaborate a little bit more about that?
So these maintaining cells, they came in different flavors because they have to take care of different needs of these cells. So they are called glia in general, but you have some that are called astrocytes, microglia. So the names are less important. But what they do is that they contribute to make the neuron to socialize with the other. So they favor these interactions. They also are responsible for helping the neuron to adapt to the changes in the environment. I mean, you have to think your brain is always changing. You are always exposed to different things. So this glia cell participate on that. And then one of the functions is also cleaning this outside. So when we were saying you are throwing garbage outside, you have some cells that are called microglia, and their job, this will be like the big trucks that are there waiting to remove some of the garbage. And at the beginning, they work well, right? It's like, when you are young, you don't have the disease, they are working well. The problem is that as we get old, as I mentioned, this cleaning decrease in all the cells of your body, including the microglia, including the support cells for the neurons. So it's kind of a like two-hit situation. Your neuron is in poor condition, and all the support system, all these fire trucks, all these ambulance, all these people that normally make sure that you are functioning as a neuron, they don't work so well because they cannot clean themselves either. And actually, in this meeting, we've been hearing a lot about how these microglia start building up a lot of undegraded materials and how that not being very helpful for the neurons because they cannot help there. And same thing for astrocytes. So there are specialized cells, but they are all necessary for the support of the neuron.
Well, yeah, you touched something about, this is a conversation about bad cleaning. So the cells are not able, for one or another reason, chaperones of the container, to degrade those proteins that are not working well. And that is involved, as you said, like Alzheimer and neurodegenerative disease. But is there any kind of pathology that is involved in cleaning too much?
Yeah, so that's a very good question. And actually, that's something that has worried me for a while. I think now I understand a little better because when autophagy was described, this cleaning process was described in the 60s. And actually, the Duke got the Nobel Prize. But then in 2016, Dr. Oshumi got also the Nobel Prize for the molecular discovery of autophagy because he discovered the genes. Um, that brought a lot of interest. Is like, well, now that we know the genes, we can activate this cleaning. Let's do that. But going back to this idea of selectivity that we talk about, the broken chair and the good chair, the problem is that most of the interventions that has been developed so far, because we need to know more molecular details, have been trained to enhance. Let's put more cleaning, recycling containers, and hopefully, and that will clean. So there are different ways to do this cleaning, the same way that at home you have the broom, you have the vacuum. So some of them are very selective. And actually one of the ones that I discovered as a graduate student is very selective. So that one we know that it can never be too much because the way that it works, you need the chaperone to identify the broken chair. So this system will never touch the broken chair unless that the chaperone bring it there.
So that is saying that one.
That one.
And,
So
and I'm going to do it
there is
fast.
a particular flag and everything
Yep.
is fine. But there is another way of cleaning that is a little more massive, that what you do instead of, having the container there, taking out wherever the chaperone fits, what you do is you create kind of a wrapping bag around things inside the cell, trying to take the garbage and then bring it to the garbage container. But imagine a big bag with an open mouth taking things. Of course, you take the broken chair, but you might also take the good one because it happens to be next to it or bound to it. Imagine those chairs that assemble one to the other. So you only want the broken one, but everything is coming along. So that's the one that when it's activated too much, can cause some diseases. And there are some developmental disorders that what happens is that because you are eliminating things that you need. Imagine, for example, mitochondria, just to move a little from the chair. So your mitochondria is the powerhouse, is the one producing the energy inside your cells. If a mitochondria is bad, you want to eliminate them because it will create a lot of free radicals and a lot of damage. But if you eliminate the healthy ones, your cells are not going to have the powerhouse there. So they are not going to have energy. So they are going to become energetically deficient and they can die. And there are some examples that you can induce experimentally if you activate much that particular type of autophagy, that might not be safe. But, the field is being very responsible and very careful with all those things. So the whole idea of drug development in this field of autophagy is going for selectivity. Let's activate the selective one. Let's do something that identify that chair or identify that mitochondria, but not the other. And that's most of the development that we have done.
So that was my point, that my next point. And this is gonna be intuitive. You already said a little bit about it, but it's important to invest in science
Basic
that
science.
that look for the fundamentals and the processes that underline several aspects of our brain. Because with that, maybe you can direct, some pharmacological treatments for people that are suffering from this. And then early diagnosis is really good, but also treatments are really important. So what do you think about it?
Yeah, no, that's a great point. And actually, sometimes my family don't understand why I've been 30 years studying the same thing. And I was like, well, if you want to fix something broken, you have to know how it works first, because if not, you don't even know what to fix, right? So in our case, for example, we were working in this very selective form of autophagy. It's called chaperone-mediated autophagy. Have to say it at least once. And basically, we knew that as you get old, it stopped working or it doesn't work so well, but we didn't know why. So first, we have to characterize how does the chaperone recognize the broken chair? How does it bring into the lysosomes, these garbage containers? What is in the containers that allow the entry into the container? So we dissect each of these steps, and eventually we compare in aging, or we compare in these neurodegenerative conditions, and then we realize there is a common thing, that there is this hook in the surface of the garbage or the recycling container that is the one that is bringing it in. And as you get old, you have enough chaperone, but you don't have enough of this hook. So even you have your containers there, nothing opens because the hook is not really pulling things in. So because we identified that, the next step was like, okay, let's put that hook genetically, because it's easier, in an animal model and see what happened. And what happened is that they are more protected from neurodegeneration, they live longer, they are healthier. So that gives the rationale to design drugs that increase the amount of hooks. And that's actually what we have done. So tomorrow I will be talking about some small molecules that we develop that are able to put more hooks into these containers. And those are, molecules that we hope they will help in neurodegeneration, and that's why we go for the garbage removal rather than what is the cause, what is the mutation, because it's common for many diseases. I mean, in many diseases, the protein can be A, B, or C, but at the end of the day, if you are cleaning it, you will not have a problem. So this kind of approach should be valid for Parkinson, Alzheimer, as far as the problem is in these recycling containers.
So the more we know, the more selective can we be for a specific target.
And
Yeah.
basic research is essential for that. And it takes 30
Absolutely.
years. It takes 30 years, but then you go into solid steps.
You said that, of course, and this is obviously intuitive, that aging is the most risk factor. But another risk factor that can be, taken into account in these pathologies.
Yeah, nobody's gonna like it. So the problem with autophagy, if you think about autophagy and cleaning, because I mean there are multiple risk factors. I mean in pathologies and in aging in general, but an important one is nutrition. It's not all about drugs and development of pharmacology around the diseases. I think nutrition, the way that we change in our society, that you are continuously snacking, is not good, because as I say, you need the incentive to clean. So for example, in your liver, in between lunch and dinner, if you don't snack in the middle, the liver will activate autophagy, because it's like it needs energy and it has to provide to the other organs. So basically we are continuously eating, so that's not good, and we don't leave time for this autophagy to happen. And then the other thing, and this is particularly important in the brain, is something that is free, that is sleep. And we don't evaluate this. I mean, we don't give value to sleep. But I always put the same example. Imagine that you have a store, right? A supermarket. So if you have everybody in the supermarket, that's not when you clean. I mean, you do selective cleaning, like cleaning in aisle 5 because there is a spill. But the real cleaning you do when you close the supermarket and then, you start
Stop work,
cleaning.
clean.
Yeah, so just stop. So, neuronal cleaning during the day, I mean, it happens, But the really in-depth cleaning happens when you go to sleep, because it's not that you don't have activity, but the activity is way less than now that you are trying to figure out what I'm explaining, right? So your neurons will go a little more relaxed. And the problem is that we sleep every time less. And then because we stay late, because we have to finish this project, you are snacking because you don't want to fall asleep. So you are killing autophagy in two different ways. And actually, I'm exaggerating, and that's not the only reason why sleep is important. But there were beautiful studies done at NIH actually showing that if you deprive young animals from sleep, they develop amyloid plaques. So this is a very serious problem. And in that case, of course, it's a variety of proteins, because it's like whatever should have been eliminated is not a particular mutant one. But that tells you how important is this sleep and this cleaning.
That's partially good because we cannot control aging. It's a natural process in life, but we can control at least partially our nutrition and our circadian rhythm. So this is good, and also that translate into the society, because the more there are like some division in society, that some are healthy, some have resources, so you can sleep well, you can have a nutritionist in your diet, whatever. But some of those population that are risky population in several ways, they might have those pathologies as well, because have bad absorption, malnutrition, all that stuff. So it's also important to build bridges between science and politics and society, right? So you can, because at the end of the day, treatment, it's expensive. So the more that you prevent, the less you're going to spend on that.
Correct. Yeah. No, for that, I mean, and you said we cannot stop aging, but we can change the way that we age. And that's my whole theme here, because it's like, we should be more aware that there are things that you can do, and they are very democratic. They don't cost money. Like, separating, I mean, you can eat the same thing that you eat, but just separate the meals. It's like, don't be continuously snacking, have the same calories, whatever you eat a day, but just separate at least like, a couple of hours so your cleaning happens. And sleep is really very democratic intervention, right? So I think that's part of the process, that, yeah, eventually everybody wants the magic pill, but not everybody is going to have access. And I'm not so convinced that it's always good to have just more pills to our elder population, right?
So I'm gonna pause a little bit, because you are gonna have a I think gonna be an amazing talk, because this was an amazing conversation, and you are gonna be the Marty Balk Lecture Award that recognizes outstanding contribution by women scientists. I'm talking about diversity, that we're gonna touch a little bit about in a few seconds. So it's gonna be here at the International Neurochemistry Society, and what are you planning to talk about in this session?
So the first thing is that I'm extremely honored for this recognition. And again, I always say it, I mean, I'm just the storyteller. The recognition goes to my lab and the people that have been working with me for so many years. But it's particularly meaningful that is for women in science, especially at this time in history, because, the support of the Society for Women has been always very evident. And also having Martha inspiring. I mean, she was one of the discoveries of neurotransmission, and we were talking about these neuronal connections and how they communicate, and she was one of the founders of that field. So just having her name even close to mine is already very meaningful, and I'm very touched for that. And then basically what I'm going to talk a little about cleaning, because I only know about cleaning. And I always say it, I'm not a neuroscientist. I'm a cell biologist by training. So I'm a cell biologist playing with neurons. And we have discovered that if we can modulate some of this cleaning, if we can enhance this cleaning in experimental models, that seems to slow down some of these neurodegenerative conditions like mouse models of Alzheimer, of Parkinson. So I'm going to be talking about that. And of course, I'm going to be talking about all the very basic biology that went into discovering the molecules that we can now target.
Well, I cannot avoid to ask you this, because you have this award that is about women in science, groundbreaking, findings in your area, but also diversity. So diversity, it's important in science, like people that is in your lab doing science, but also in clinical trials to have diverse populations to evaluate them.
So we always talk in science, we talk about diversity from those two angles. As first, the people that are doing the research. And I think it's extremely important to have diversity in a lab. And that's some of the things that I love of science. It's like you have in your team, you have people from all places, all ideologies, and it doesn't matter. We all have a common language, but very different ways of thinking. And I think that's what enriches science. The way that I might formulate my questions and my hypotheses, completely different from my students, from my postdocs, and then you engage into this discussion and who is right, who is wrong, you try to prove it. So I think, and you know, the way that you think is very much related to where are you from and who you have been exposed to, right? So I think diversity in research is very important for the team, but also diversity regarding the research itself. And it sounds very silly that it's like 2025 and 2025, and we still don't know many of the difference between just not talking about diversity in the big sense of the word, but even males and females, like women and men. It's like most of the drugs have been developed, tested in male mice, because that's what we do, because it's cheaper, because then you don't have to worry about many other things and hormones and pregnancy. And that's a big mistake. And this meeting has been really exciting on that, because you can see very young, bright scientists embracing this idea. Is like, guys, you guys are wrong. This neuron from a female, very different from a neuron of a male. And actually something that I'm going to touch tomorrow too is that even the cleaning is done different in males and females. And that probably tell us about our risk for disease. So that's just in the everyday male-female thing. But then when you think about populations, I mean, I think the big next step for us as a lab and as a field, for the field of autophagy, is trying to understand a bit more about human autophagy. I mean, we have done really considerable amount of work in yeast and flies, mice. We are starting to do studies in populations, and actually I have very good collaborations with Spain. And we are trying to see the influence of the Mediterranean diet on autophagy. And you can see that depending on the origin of the person, even the basal level of autophagy, probably between you and me, is completely different. And you have to understand that if you are going to treat and you want to enhance or modify. So I think having diversity, the risk factors are very population dependent. It's not the same one. So, and that's the beauty of having science being universal, is that I can talk with somebody in Tokyo and somebody in Colombia, and you can still understand what you are talking, but you are working with different populations. So you can enrich each other and see what is universal and what is very community or area specific.
And it takes time, because you need to have, like, policy science sometimes work different in different parts of the world. So, but it's necessary. Right? Got it. So congratulations again for that award. We are really happy to hear that tomorrow, I guess.
Mm-hmm.
Yeah,
Tomorrow
nice.
morning.
So moving a little bit about forward in this conversation and talking a little bit about who Ana Maria is outside the lab. So what do you want to do outside the lab? What do you like to do? What are your hobbies? What do you want to spend time on?
Yeah, so my outside the lab is usually an airport or a conference center. So that tells you how much life I have. But it's my choice. I mean, I love this. This is what I do for a living. Like having the opportunity to today talking like somebody like you. I mean, this is enriching my person, and I that's what I love. But in general, because I travel so much, like really crazy amount of travel, all conferences. I love to meet people from different places doing science, because as I say, you have a universal language, but you still learn in such a different way. When I'm not in the road, I like to be home. I am very boring on that, but I just like to I enjoy family time, and I enjoy my cats. So that's pretty much it. I'm the old cat lady.
Love it. You need to take rest. You need to have time for yourself as well. So that's important. We really like it. So, and talking about travels. So, the next ISN meeting gonna be in Kyoto, in Japan. So you're gonna be part of the chair of that. So
Yeah,
tell
I
me a
have
little bit about
the
it.
privilege to have been selected to be the chair of the scientific program. So we have a wonderful committee of experts that will be the ones judging the different applications, I hope, for many people from the society. So the symposiums that you see during the meeting, those are all originate from the creativity and the interests of the members of the society, like students, postdocs, investigators, and basically you can prepare an application if you are interested in a particular topic related to neuroscience and neurochemistry. You can prepare an application just telling what will be the theme and what will be the people that you will invite to present their work because you like their particular area or because you think it's something that the members of the society should learn. Doesn't need to be necessarily hardcore neuroscience, like even areas from outside always enrich the society and the science of the members. And the idea is that we hope to receive a lot of applications. You are never too young or too old to put one of these applications, so go for it. And then basically I will count with the tremendous help from the whole committee reviewing those and selecting whatever sounds interesting and exciting.
Well, so far this meeting in New York has been like really, really good, really exciting. But it sounds exciting as well, the upcoming one. So can't wait to be there. And it seems like it's a nice city. We're gonna have some
Kyoto is
episodes
a
there.
wonderful city. Yeah, I can tell you.
So the message: eat well, sleep well. You're gonna age well, and your system gonna clean well.
It's all about cleaning. Like my mom says, in a clean house, everything works better.
Yeah, that is like a Hispanic thing. I have the same. I can relate. So thank you very much, Ana Maria, for being with
Thank you,
us.
William,
It was
for
a
having
pleasure
me.
to talk with you, and see you next time.
Thank you for listening. Don't forget to follow and subscribe for more neurochemistry talks. Till next time.